It's not for me. Not my. Okay, we begin lecture. Yes. And the first, the cell is the basic biologic system. So we know that there are two main types of cell organization. They are eukaryotic cells and prokaryotic cells, and difference between them presence or absence of nucleus. So eukaryotic cells have nucleus and membrane um, and organs that have membrane. Prokaryotic cells do not have nucleus, only DNA in the center of cell, and they have no organs with membrane. Can I change this slide? Give me answer, please. Plus or minus? Yes or no? Thank you very much. So, cells that may be in group prokaryotic cells, there are two main groups. This is bacteria and archaea. You have a question? Do you have a question? Okay. So, the next. Other cells, eukaryotic cells, they are in all other organisms. So it will be kindreds of eukaryotic cell organisms with the eukaryotic cells. So it will be kindred or kingdom protozoa, kingdom fungi, kingdom plant, and animal. All these organisms with eukaryotic cells. Can I change our slide? Yes, of course, you must write it. And after that, when you will be on Kirov, and now if you in Kirov, show your teacher. So this is a big slide with very small letters, I understand. So what we have there? There are two types of cells, prokaryotic cells and eukaryotic cells. Again, two main domains in prokaryotic cells, the main bacteria and archaea. Archaea is the prokaryotic cells that are organisms that live in difficult environmental environment. For example, in area with high temperature, very high temperature, and usually they don't cause any infections. Bacterium, they live with us, with our environment, and can lead to diseases. Eukaryotic cells, again, four kingdoms: protozoa, fungi, plants, and animal, plant and animal. And they are different in their cell wall. So animal cells, they have no cell wall. They have only plasma membrane, and this membrane contain glycocalyx in outer side. Protozoa, some protozoa can have cell wall, but usually they have no. Fungi and plant, they are with cell wall. Fungi, they have cell wall with murein, plant with cellulose. Some protozoa and plants, they may take part in photosynthesis. Animals and fungi, they can't do this. So they have energy and nutrition from organic elements that surround them. Is it clear? Okay, the next slide. So, kingdoms differ. And that did I say before, you see here. So plants produce their own sugars and other nutrient molecules through photosynthesis. Fungi absorb these nutrients, and animals get food by eating. So all these cells are different. Eukaryotic cell contain organelles enclosed in membrane. And these organelles present only in eukaryotic cells. But some organelles may be in all eukaryotic cells. For example, it will be Golgi complex or Golgi apparatus, endoplasmic reticulum, lysosomes, and mitochondria. Some organelles, like plastids, have only, for example, plants. And these organelles, they need them for photosynthesis. It is easy all that I said, yes? Not difficult. We don't, yes, okay. So the next, prokaryotic cells. They have no nucleus, but they have nucleoid. Nucleoid, this is the complex of DNA with the proteins that take part in transcription and translation. So all processes, transcription, translation in prokaryotic cells are in cytoplasm and near with the DNA. So proteins that take part in these processes must be near with the DNA. And this complex we called nucleoid. Is it clear? Thank you. Thank you. So nucleoid, thank you, include circular DNA molecule and proteins. Some prokaryotic cells have not some, all prokaryotic cells, they have small size if we compare with eukaryotic cells. So main difference between eukaryotic cells and prokaryotic cells, this is their size, eukaryotic larger, absent of nucleus in prokaryotic cells, and absent of membrane organelles. Only organelles that need to biosynthesis, like ribosomes. Excuse me. Ahmad Irza Jamil, what is the question? There are all groups. Excuse me. Yes, this lecture for all group. for all groups. And table with the comparing of prokaryotes and eukaryotes. So you see size about 10 times difference between size prokaryote cell in prokaryotic cells and eukaryotic cells. Present nucleus in eukaryotic cells. DNA is not bounded with proteins histones. Histones proteins present only in eukaryotic cells and help to protect DNA from environmental factors and regulate synthesis of proteins. Now, they have no only one gene. They have a lot of genes, but the frame of reading of these sequences, they may be cross together. So in eukaryotic cells, genes located near with the genes and one sequence reflect only one sequence of amino acids, nothing more. In bacteria, it will be some different amino acids in one sequences of amino acids in one sequence of gene, because it may be different frame shift, frames of reading. Is difficult, and it will be better to ask during lesson. Your group number 105. Are you in Kirov? No. So I will try to show. Ой, не то. So in prokaryotic cells, sequence of DNA nucleotides synthesis may begin from the first nucleotide, and it will be one variant of amino acid sequence. It may start from another nucleotide, and it will be number 2, another protein. In eukaryotic cells, genes located only in one sequence, so it may be only 1 and 2 after, not together. Is it clear? In prokaryotic cells, one sequence contains information about different genes. So this is one sequence. There will be promoter and somewhere will be terminator. Several genes inside. For example, 3. 1, 2, 3. But it may be beginning from this, and you will have one gene. From this, it will be second gene. From this, it will be third gene. But sequence of first gene will include third gene. Do you understand? So can we return? Can we continue our lecture? Thank you. So the next. their division. Eukaryotic cells divide by mitosis or meiosis. But prokaryotic cells have no ability to mitosis, and they may have only simple division. Is it clear? Because they have no chromosomes, they have no centrioles. Eukaryotic cells have three components. You must know them yet. This is cell membrane, nucleus, cytoplasm. In prokaryotic cells, there are two main components. It will be cell membrane and nucleus. Is it clear? Thank you. Plasma membrane. You see, there are three main components. Plasma membrane itself, complex that will be on the membrane, or microfilaments of skeleton, and that will be in outer side of membrane. It will be like a calyx, if we will talk about animal cells. So, the first model was not correct because our diagnostic method was not good. Our microscopy was not good. So, model was not good in understanding the process that may be in our membrane. So the first model was proposed by Davidson and Daniel, and the name was a sandwich model. According to this model, there was two layers of lipids, like now we know, but they was without proteins inside, only up under and outer membrane. So, okay, it's okay. So you can see the dark line, it will be proteins layer, and two layers of phospholipids molecules between them. They are without separatists, they are without ability to movement. But location of phospholipids was correct. So tail and head, hydrophilic head and hydrophobic tail. Can I change the slide? Thank you. In 1972, another model of plasma membrane was proposed by Singer and Nicholson. And this model is correct for our time, for these days. This model good in description of function and structure, and you can see that this model is mosaic. Each element can move, and different parts of membrane include different structures, structures element, and so can take part in different functions. So the main part of membrane is a bilayer of lipids, like was in previous model. What lipids? They are phospholipids mostly. It may be glycolipids, a little percent, and it may be cholesterol. So it may be and another lipids, but they amount of very little or in compare if we compare with these three types of lipids. The next slide, yes? Thank you. So the main components phospholipids. Each of them has caput or head and a nonpolar tail, which contain two chains. The head is hydrophilic, and contain phosphor group. The tail is a hydrophobic part and consists of two chains of fatty acids. Can I change this slide? Thank you very much. So polar heads are attached to intracellular and extracellular fluid. Thank you. Therefore, they lie in the inner and outer surface of the membrane. So in microscope, we can see three layers. We know that there are two layers of lipids, but in microscopy, we can see three layers, two hydrophilic and one between them hydrophobic. Non-polar tails, they avoid water, so they will be in the intermediate part of membrane. Is it clear? Thank you. So I'm the next slide. But some chains may be different. Some of them may be with a double bond between atom of carbon and with one bond. If they have doubled bond, they will be unsaturated. Without only one, they will be saturated. It is important to understand the movement. So membrane phospholipids are unsaturated, which may be unsaturated, and it will lead to nick of tail, so it will increase fluidity of membrane. And the next, yes, the next, this is glycolipids. About 5% from all lipids in membrane. So they are important. Glycolipids have a sugar group, and often they are located in outer part of membrane. They are important for reception, for attachment. I didn't ask. Can I change this slide? Did you have enough time? Total membrane lipids include else 20% of cholesterol. Big percent for membrane and function of this. Cholesterol help to stabilize membrane. So its hydrocarbon rings are located between the tails of phospholipids, what help to stabilize the membrane. and reduce movements. Is it clear? Thank you. So some parts of membrane more fluid according to the structure of tails of phospholipids. Some parts are stable and helping with cholesterol and structure of tails of phospholipids again. Now about proteins, very important part of plasma membrane. They may be different. They may penetrate membrane or located only on the surface. So you can see different variants of penetration. When penetration will be only in one side, for example, you see protein number 4, or protein will penetrate both layers, like protein number 3. You see location of float membrane, float freely proteins. It may be outer part of membrane or inner part of membrane. If they are located in inner part, they will take part in supporting of shape or in chemical reactions inside cell. Proteins that located in outer side help to reception and to connection of cells together. Is it clear? Thank you very much. And again, model of membrane, cell membrane. You see all components now. So you see phospholipids with different formation, different structure of tails. You see cholesterol for stabilization. You see glycolipids for reception. You see integral protein. Integral protein. This is protein that can penetrate membrane, and this protein, big yellow protein, integral protein, it will be transmembrane protein because it penetrates and outer and inner part of membrane, and maybe peripheral protein. You see this protein located in inner part. So while we use a lot of time for understand structure of membrane, the first we must understand how it possible to transport some elements from outer side to inner side and opposite from inner side to outside, and it will help us to understand in future in the second course how it possible all functions that can take part all cells, each cells in our organism. So according to the structure. Again, about integral proteins. So we said it may be transmembrane proteins, and very often with these transmembrane proteins are linked peripheral proteins from difficult functions. For example, signaling. Cell have signal from another cell. The signal will be transferred from peripheral proteins to transmembrane protein. After that, again to peripheral protein or integral protein, and after that, this signal will go to the nucleus, and it will lead to biosynthesis of another new structures. If you try to tell me something, I hear only noise, not your voice. I'm sorry, I can't hear you. Do you have a question? Thank you. So the next slide, again about integral proteins. These proteins always must have an hydrophilic and hydrophobic sites to locate inside membrane. Excuse me? You talk with me about all lecture. Are you? Madraza Jamil. Did you ask me about all lecture? Can you explain your question? Are you talking about all slides in all lecture? No, all lecture I will not give you because if you didn't listen me, you don't understand what happened in this slide. I can give some slides that can be understood without my explanation if chief of the course will ask me. So if he will ask me, I give him this information and he will share for all groups. Only this variant. So Amir can find me and ask. Thank you. Transmembrane proteins. Transmembrane proteins and their function. So the main function is transport. But there may be two variants of transport. It may be like channel and small molecules, like water, for example, may go through this protein. But more often, small substances will be attached to protein and protein will transport, will move them through the membrane. And this protein will cross this membrane with them. Next slide. Yes? Or you need time? Thank you. So transmembrane proteins, the next function. It will be enzymes. More often in signaling. So enzymes for transfer signal from one cell to another, but maybe some metabolic process, chemical reactions too. Some transmembrane proteins are receptors. For example, for hormones. Next slide, yes? Thank you. Thank you. And you see another picture that can help you function of proteins. You see again that it may be transport, reception, and in the down angle in the left, you see that it may be supporting of the function by attaching to the cytoskeleton. Excuse me. Nobody can hear me. So it's mean something happened with your computer. Thank you. Thank you. So I will continue. Thank you. And peripheral proteins, they may. Ahmed Maher, I don't know what did you write, I'm sorry. These preferences can be freely bound to integral proteins. Like you see in this picture, in the outer side, it will be E. I understand something with his computer. Or in the inner side, you see these proteins take part in process of phosphorylation. What else? They can support the membrane from in cytoplasm. And so according to these proteins, the allocation, it will be different shape of cells or ability to constriction and migration. So surface identity marker. So you know we have immune system. We have immune system. And some proteins on membrane will have certain shape. Our immune system will find this shape and will recognize this is our cell, we will not destroy it. But if, for example, another cells will have another, for example, protein, another shape, our immune system will find another receptor. It will be signal to destroying this cell because another non-correct protein. Is it us in your question? Hannah do Eugen, I'm sorry if it's not correct. Do you have answer or you need explanation else? Thank you. So continue. We said that proteins and lipids are free. They are not don't connect each other together. So plasma membrane can be like a fluid. What is direction of movement of these molecules? So it may be lateral to the place of neighboring molecular. It may be rotation and it may be transverse movement like flip-flop. But some parts of our membrane can't change their structure. This is lipid rafts. They include constant saturated phospholipids, sphingolipids, and cholesterol. More often, this is part for reception. Is it clear? What is it, lipid rafts? Thank you. So the next. About function, yes, that more often platforms for receptors. And these parts very stable. Now the consequences of that we said. Properties of plasma membrane. It will be questioned on the exam, on small exam, during final exam, and nobody of students, it was 2 final exams, nobody can answer this question. Please be careful. Work with attention. These important parts of your study. So the first, fluidity of the plasma membrane. Due to structure of lipids and the ability to change their position. The second, plasticity. The membrane can change its shape without losing, I'm sorry, double O must be, losing contacts. Next slide. Okay. So you will take exam card and you will read properties of plasma membrane. Your answer must include properties, for example, 1, fluidity of the plasma membrane, and explanation of this. Why plasma have this property, fluidity? Because lipids may be unsaturated and saturated that lead to different mobility, and that's why lipids can change their position. So it will be explanation of fluidity. The next, you will explain what is it, plasticity. That membrane can be repaired. Excuse me, not this. That it may change their shape. About reparation will be the next slide. Do you understand, Ahmed Salah Mustafa? Thank you. The next property that you must know, the ability to self-close and selective membrane permeability. The first of this. Membrane spontaneously closed in a circle shape when it is damaged. This is their property. More often it happens with the help Golgi complex or Golgi apparatus function. Selective membrane permeability. The function of the plasma membrane is a barrier. So some substances may go through membrane, some of them can't. Some of them will use energy, some of them will diffuse without ATP molecules. Is it clear? Thank you. The next function. It will be structural function, barrier function, metabolic function, receptor function. Structural. So components of all cell organelles except ribosomes and centromeres, they have the same structure. So it will be compartmentalization of cell. Each compartment will take part in different functions of cell inside cell. Barrier, it protects from against external factors, for example, bacteria or viruses. And metabolic, you saw that a lot of proteins may take enzyme function, and they are located in the inner part or outer part of membrane. Receptors, you saw it usually proteins that connected with lipids or sugar that connected with the bilayer of lipids, bilayer of lipids. Is it clear? Can I change this slide? Thank you. So, this is very important that our membrane can recover from after dangers of the destroying. The next layer, outer layer, that located outside, this is glycocalyx. This layer present only in animals. So glycocalyx is the outer coating of eukaryotic cells. Yes. What is the question? Question, please. If there is a little space between two ends of membrane, they will be connected according to the chemical forces and electricity forces. If there is a big rupture, wait a second. If will be big distance between elements, so like this. Golgi complex will produce vesicles that will be connected with the membrane, so they will reparate this part of cell. Is it clear? Thank you. So we can continue. Yes? Thank you. So each type of cell has a different sugar in its glycocalyx. And it will be biological markers for each cells. For example, for immune system or for sperm that must recognize egg cell. Can I change the slide? Is it clear? Thank you. Thank you. Eukaryotic cells are divided into compartments, we said it, by internal membrane, and each compartment will take part in each one function. So it will be some units of cell that will function like one organelle, for example, endoplasmic reticulum, or lysosome, or Golgi complex, or Golgi apparatus, yes. And now, some of you, that students that in Kirov, yesterday and today had some small exam. Found structures and gave them name. So for the students who didn't do this text good, look at this picture. So Golgi apparatus. This structure never have boundaries with nucleus. You see, this is smooth, this is roof, and the plasmic reticulum, they always connected with the nucleus, and sometimes they will have connect with the outer membrane. Don't confuse these two organelles. About mitochondria, you can distinguish them due to having crists. So they have two membranes, and inner membrane create crists inside. Lysosomes, they are vesicles that are located near with Golgi complex, and they may be in another part of cell, not only near with the complex, but it will not be a mistake if you will confuse lysosome and vesicle, because they are was produced by Golgi complex, so it's not rough mistake. And centrioles always pair organelles, so they must be together near the nucleus or in different poles. They are like cylinders, not like vesicle. So main organelles that you may find in our picture, we discuss. Do you have questions? Do you? Thank you. So about each organelle, endoplasmic reticulum. Endoplasmic reticulum, yes, Amir. Endoplasmic reticulum is a system of channels located through the cell and associated with the perinuclear space of the nucleus and the cavities of the Golgi complex. Its wall is a plasma membrane. Amira, you have a lesson in this time. If it's so, call me after today in the evening after 6. Okay? Так, the next slide. Can I change this slide? Thank you. Rough endoplasmatic reticulum has ribosomes on its membrane. It participates in the biosynthesis, thank you, biosynthesis of proteins, which are then transported to the Golgi complex in Russian variant or Golgi apparatus in English variant. The next slide. Thank you. So ribosomes are attached to the outside of endoplasmatic reticulum. You can see on the right side. On the left side, you can see ribosome. Ribosome, this is a complex of RNA, ribosomal RNA, and proteins. It consists of two subunits, small subunits and large subunit. Between them will go process of translation. So number 2, you see, it will be matrix RNA. And number 3, transport RNA. So they work together to make up acids sequence that will be protein in the future. I'm sorry. So after that, this protein will go inside between two membranes inside endoplasmic reticulum and go through the endoplasmatic reticulum to the Golgi complex to formation. Third level of structure, fourth level of structure, or for transport in the another part of cell or to the out of cell. Is it clear? Thank you. The next variant of endoplasmic reticulum. This is smooth reticulum, which thank you, does not contain ribosomes. So what is the function? Function is the synthesis of lipids and sugars or carbohydrates. It take this will be that we discussed before. Attachment one cell to another according to same structure of proteins. So proteins can help to connect to different membrane of different cells. If it will be a lot of cells, it will be organelles that can attach to each other. Do you understand? Or you need that I draw this? Thank you. Oh, excuse me, please. And what else can be synthesized inside? It will be steroid hormones and some substances that help in detoxification. For example, in liver cells. Golgi complex or Golgi apparatus consists of vesicles, tubes, and sacs. This complex we call dictyosomes. They are main elements of the complex. Dictyosomes, they are like closed sacs that are thin in the middle and have extensions at the ends. These extensions will form vesicles to transport of some substance, or for formation lysosomes for digestion, or vacuoles in plants. Is it clear? Thank you. Functions of this organelle. So it will be sorting and packing of substances that was synthesized in endoplasmic reticulum. Formation of complex compounds like lipoproteins, glycoproteins, assembling plasma membrane that we discussed before during damages. Formation lysosomes and other variants of vacuoles, and participation in the secretion of some substances. For example, endocrine cells. Is it clear? Thank you. Lysosomes. Thank you. Lysosomes, they may be primary and secondary. Primary, formed in Golgi complex, they are spheric, and inside them we can find hydrolytic enzymes for digestion. Secondary lysosomes, they contain digested substances, so they are formed after fusion with endosoma. Next slide. Thank you. Thank you. Functions of lysosomes. Breaking up substances passed into the cell by phagocytosis for digestion, and destroying impaired structures and organelles of the cell. So it may be component of apoptosis. Apoptosis, this is programmed cell death. Is something wrong inside? For example, viruses that inside our nucleus, or some damage after trauma. It may be different reasons that can lead to death, programmed death, and organelles that take part in this will be lysosomes and mitochondrials. About mitochondrials, we will talk in the second course. About lysosomes on parasitology, I think. So the next slide. Thank you. Another variants of vesicles, for example, pexisomes, they are formed in endoplasmic reticulum, and their enzymes oxidize amino acids with formation of peroxide. And glycosomes near will be rough, after that will be smooth. So the first more often will be rough because ribosomes will present and in outer part of nucleus membrane and in roof. So more often will be roof endoplasmic reticulum. But some cells may have endoplasmic reticulum smooth, but not often. Is it clear? Thank you. The next, glycosomes. They are formed in Golgi complex, and their enzymes transform fats into carbohydrates. Very important function. So the next slide. Mitochondria. They are shaped like roots, but they may have another shape, like filaments, for example, or like granules. It depends from cells inside which we see this mitochondria. Sizes may differ too, from 0.5 to 7 microns, and the number of these organelles may be different again. It's depend of function of cell, how many energy they need. So then more energy need for function of cell, then more will be mitochondrials inside. For example, 1 hour, or I'm sorry, 1,000 or 10,000, only 100. Next slide. Thank you. So the structure. You see there is two membranes inside. Membrane inside will formate crisps for respiration. Abdulkadir Hassan, I'm sorry. On the noise, I can't hear your voice. I can't hear you. I think will be better if you will write. Nothing that can I understand. Excuse me? I don't.
عبد القادر يا عم اقفل المايك.
Okay. Thank you very much. The inner membrane forms crisps for formation ATP molecules. And gap between them perimitochondrial space. Function of mitochondria. The main function is the ATP synthesis for energy. So now we can return to mitochondria. Yes? So ATP synthesis. Some specific proteins may be created inside. For example, steroid hormones. And participation in apoptosis. You may find more functions that I tell you, but if you will tell all of them during an exam, but don't tell these three, it will be not good. So these three main functions must be always. Others, if you wish. If you wish to say, of course, it will be great. Under inner membrane, it is a matrix that contain enzymes. I'm sorry, I think about another things. Enzymes for the oxygen stage of energy exchange. These enzymes will be synthesized in outer DNA. So in matrix, there is a DNA, but not one. There are copies of DNA from 2 to 10 copies inside one mitochondria. I said that all lecture I will not send because some slides need my explanation, but about 30 slides you will have. If Amir will come and ask me about this. So, some proteins. Daniel, I can't understand. What do you want? I said some of slides I will give. Okay, thank you very much. If you need on site, there is you can find my mail. So all questions you may write on my mail, but before 11 o'clock in the evening. Please don't write me at the night. I won't sleep. Book only in our library, but if you want, I can give you next lecture some books that will be useful for understanding. You can find them in internet. Okay, one book very good. I can give you author and name of this book, and you will find it on internet. Okay. Thank you. But next lecture, I have no information with me now. Thank you. So under the 9, about what? This slide we discussed yet. Return to common organelles that present in all eukaryotic cells. Again, they are smooth and rough endoplasmic reticulum, Golgi complex, mitochondria, ribosomes, lysosomes, and peroxisomes, microfilaments and microtubes, and centrosome or centrioles. But plant cells have no centrioles. The next slide. Amir, I don't understand you. Who all? They are not in Kirov. How can he come? Only 2 groups. So, we have no time for this exercise. Membrane-bound organelles. We said that there may be double membrane bound and single membrane bound. We must classify our organelles. So, 2 membranes have nucleus, but nucleus is not organelle. Nucleus, this is main component of cell. Please don't call nucleus the organelle. Never. This is main component. Organelles, only mitochondria and plastids in this slide, okay? Again, so if will be question, what is the structures inside cell may have 2 membranes? You will say nucleus, mitochondria, plastid. If will be question, what organelles have double membrane? You will answer only mitochondria and plastid because nucleus is not organelle. Nucleus is the main component of cell. Okay? Thank you. So these 2 organelles, mitochondria and plastids, contain DNA as genetic material and ribosomes. They may replicate their products from this genetic material without help of nucleus. So they will be autonomic organelles because they may create their own ribosomes without help of nucleus. Next slide. Thank you. The cytoplasm. So we discuss only organelles, but they are located inside cytoplasm. This is jelly-like formation, fluid formation that contain microfilaments, and together microtubes will formate a structure that we call cytoskeleton. And about cytoskeleton, there are several variants of components. It may be microtubes, microfilaments, and intermediate filaments. What is the function of this cytoskeleton? It will be reinforces the shape of the cell, acts in movement, for example, intracellular transport. They take part in division, and they are polymers that contain monomers, proteins. Next slide. Or you have questions about this? Okay. Yes. Again, cytoskeleton. This is a complex of some structures. They may be three main structures. It will be microfilaments. They will be intermediate filaments, and it will be microtubes. They are different in chemical structure, and they take part in different functions, such as movements of cell, supporting the shape, transport. You see in angle of the slide vesicle that will go along the microtube with help of motor protein. So this microtube will be like a road for this vesicle. And it will be construction of some cells like muscle cells, and it will be help in division of cell. Do you have my answer? Oh, Hannah, I'm sorry that your name is so strange in my variant. It's difficult for me, excuse me. So the next slide, yes? Thank you. So you can see three variants of elements. It will be microtubes. We said it will be transport and supporting the shape. It will be intermediate filaments. Microtubes take part in division of cell. Intermediate filaments, they help to support form, and microfilaments. They are very important to formation some protrusions like fibrils and for movement of cell, like do this amoeba. Next slide. I think it will be last slide. So you see that microfilaments, they are monomers that compose these microfilaments, like globules of actin, and they are formed fibers. Intermediate filaments, the same, but another component. It will be fibrous proteins. And the next microtubes, you see there are two variants of monomers, alpha globulin and beta globulin, tubulin. So different shape, different structure, different functions. And there a little part of our lecture we will finish next time. And next lecture will be about nucleus, DNA and biosynthesis of protein. So again, please, that student who not in Kirov, please don't forget, send your homework on our side. Without this, it will be that you miss our lesson. It means you will have отработка in the end of the semester or end of the year. It means you will not enough time to prepare to another lessons. It means that you will have retake of exam. Seriously. Your third year students, the first 2 year students who didn't do that I say, they until now in the first course, so they can't finish biology. Don't forget, post your work. No, lecture you will show after. So, all excuse me, you are from what group? I'll say it. One zero three. You are not in Kirov, so you have no test on biology. You will do this test when you will be in Kirov. It will be official days when you will come on our department and will do all written works that you must. Thank you. Thank you for your work. Thank you for your attention. See you next week. Right now, yes. When you will go to the Kirov, when you will go to our department, you will have the same tests. Thank you. Thank you. Goodbye. I wish you a good day. Have a good day. Goodbye. Bye. Thank you. Thank you. Goodbye.